Good evening, everyone, and welcome to the Secret Science Club. I'm Dorian Devens, and I think Margaret Mittelbach is there.
Hello, hi there.
Okay, and now it is our great pleasure to introduce to you the new president of the Dana Foundation, Dr. Caroline Montojo.
Thank you very much, Dorian, and welcome everyone. I hope that you all are enjoying your gray matter mocktails. I hope to join you in getting a mocktail myself shortly. It is such a pleasure to be with you all this evening. This is my first Secret Science Club lecture, in fact, and we are looking forward to a great talk by Dr. Yasmin Hurd on the important topic of addiction and addiction treatment as part of Brain Awareness Week. So for those of you who are not familiar with Brain Awareness Week, it's a global campaign coordinated by the Dana Foundation to foster public enthusiasm and support for brain science. Every March, our partners, such as the Secret Science Club, host activities that highlight the wonders of the brain and the impact that brain science has on our everyday lives. So if you're interested in learning more about how your brain works, about human behavior, about the human mind, I strongly encourage you to visit our calendar on brainawareness.org to find events that are planned all week long. And so now, without further ado, let's turn our attention to tonight's talk and our wonderful speaker, Dr. Yasmin Hurd. Dr. Hurd is a professor of psychiatry, neuroscience, and pharmacological sciences at the Icahn School of Medicine at Mount Sinai. She's also the director of the Addiction Institute at the Mount Sinai Behavioral Health System, the Ward-Coleman Chair of Translational Neuroscience at Mount Sinai, and a member of the Dana Alliance for Brain Initiatives. She's conducted pioneering research on the neurobiology of opioid abuse, the neurodevelopmental and cross-generational effects of cannabis, and potential treatments for opioid addiction. And with that, I'd like to now turn it over to Dr. Yasmin Hurd. Dr. Hurd, thank you for joining us tonight.
Caroline, thanks so much for, the honor of being here for Brain Awareness Week with the Dana Foundation and with the Secret Science Club. It's great to bring together so many different people with this important topic. And as I said, it's great to be that person giving the message today about the brain. And let me start sharing my screen because I think we're going to have a long night with a lot of questions, hopefully. So as everybody mentioned today or tonight, I'm going to talk about the vulnerable brain. I was asked to talk about, in one sense, pathways to and from addiction. And I start off with the fact that, to me, the US is definitely a country of drugs. We have a lot of people who, with recreational use and misuse, and over 30 million people who have a clinical diagnosis of a substance use disorder. There are many drugs that people use, and substance use disorders are, one of the things here again, substance use disorders are really, it's challenging always to say because people never believe the amount of the burden, both on an economic and a personal level, that substance use disorders have in our society. We have nearly $1 trillion of the intangible loss of the quality of life because of substance use disorders. It's also challenging because so many people require help, but so few receive it because of a lot of these issues, not just the economic burdens that I emphasized before. We are in an epidemic of overdose deaths, but the stigma of addiction still makes such a huge impact. And altogether, the healthcare system is really under siege. So, we've spent a year, all of us, in extreme stress with COVID. And that has actually also impacted on, the social isolation and drug use. And we have today, from the last year, looking at the national increase of drug use associated with COVID and the stress and the social isolation. And it's not just alcohol. Alcohol is also increased, but so are these drugs such as the opioids, fentanyl, heroin, and cocaine. And indeed, people who suffer from a substance use disorder during this time of COVID, their odds of developing COVID-19 is actually quite high, the odds compared to those who don't have a substance use diagnosis. And they're often more hospitalized due to COVID and will die. So what is addiction? I mean, and we can go back and forth about our society's definition of it. It's a complex disorder. Webster gives this long definition of a chronic, compulsive, habit-forming behavior, and the National Institute of Drug Abuse talks about it as a chronic disease, characterized by drug-seeking behavior. Many people think of addiction in a very colloquial manner, but it's a clinical disorder where it's a substance use disorder with clinical diagnosis. And it's not for you guys to read the criteria that sets for what gives you a diagnosis of substance use disorders, but it comes back to aspects of impaired control, social impairment, risky use. And altogether, I think that what everything has in common is this aspect of chronic drug seeking and inhibitory control and craving. I'm going to talk a lot tonight, because it's Brain Awareness Week, about the neurobiology of addiction. I will start off with a very generalized overview of the brain, of what we know. And we actually know a lot about the neurobiology of addiction, but we still need to know a lot more. Some of the the classic areas that people study and know about, even people on the street, about addiction is, for example, a brain region called the nucleus accumbens, which is really important for reward and goal-directed behavior. The dorsal part of that, the nucleus accumbens, the dorsal striatum, is important for habit formation. Our cortex, in particular the prefrontal cortical regions, the orbital frontal regions, are important for cognitive control, motivational drive, cognitive flexibility. And the amygdala, which sits in our temporal lobe, is really important for emotional regulation. And hippocampus, not shown on this figure, but important for memory. And a key part for a lot of this is a brain region in the brainstem called the ventral tegmental area, the VTA, which is really important for a number of the neural circuits related to the reward pathways. And on a neurochemical level, a lot of the drugs and the substances that are abused, they impact on multiple transmitter systems in the brain. So they impact on the classic transmitter system that many people know about for addiction, such as dopamine, but they impact also on the transmitter systems of glutamate, which is the primary excitatory transmitter in our brain, or GABA, the prominent inhibitory transmitter, but also the opioid neuropeptides, opioid receptors in our brain, and the endogenous cannabinoids. So drugs of abuse, they leverage our neurobiology, our endogenous systems to conduct their, to induce their rewarding effects and leading to addiction. But what many drugs of abuse and many substances of abuse share in common is that most of them impact on this dopaminergic system, especially this mesolimbic system, that VTA to nucleus accumbens system, where when dopamine levels go up, it's associated with, following drug use, is associated with euphoria. And when it goes down, it's usually associated with dysphoria. And this activation of the nucleus accumbens has been associated with, as I said, reward and dopamine. But one thing that is also clear is that addiction is not really a disorder of reward. Reward may start the process, but we see this changes in synaptic plasticity, how cells talk to each other, is a key aspect of the neurobiology of addiction. So a big question for all of us is that we know that many people use drugs of abuse, yet still there are only a certain percentage of people who develop a problem. And so the question is why? And can we learn anything about the brain in this context to give us insights into the disorder and even potential treatments? And one of the things that we know in regard to addiction risk, well, we know many things about addiction risk, but clearly neurodevelopment is important for addiction risk. I'm going to come back to that, especially later in my talk. We also know that environmental factors, so stress, early drug exposure, are also critical to that. Genetics does play a role, and together with the environment, interacts on what I'm going to talk about, epigenetic mechanisms. And together, genetics and epigenetics impact on behavioral traits and psychiatric comorbidity that leads to these traits relevant to addiction risk and the substances that people consume. So I'm going to go over each of those a little bit and then focus in large part on some of the developmental studies and also on getting out of it in terms of treatments. So in regard to genetics, I mean, it's known for a very long time, obviously, that each drug has a different sensitivity to people becoming addicted to it. So, nicotine is a highly addictive drug, for example, and heroin. But heritability in terms of, when they look at twin studies, which substance has a greater heritability? And you can see like drugs such as cocaine and opioids have a significant heritability for their addiction. And if I use as an example the endogenous opioid system and opioid use disorder, for example, we know that there's a moderate to high genetic load for opioid use disorders. And we know also that the receptors where heroin or morphine, its metabolite, binds in the brain, for example, these called a mu-opiate receptor, which is important for mediating reward, positive mood, and euphoria. And when we started to understand that there were genetic polymorphisms in the genome that were functional and that could impact on the sensitivity, for example, of this mu-opiate receptor, it was clear that this might have a role in perhaps opioid use disorder. So when we studied this mu-opiate receptor, and I'm going to go back in one sense in terms of just a single nucleotide polymorphism of this mu-opiate receptor, basically every 300 bases or so in the genome, there is a polymorphism. And this one turned out to be functional. And when we looked in our patient, or not patient population, these were people who had died, but in our heroin overdose population, nearly 90% of them had this polymorphism of their mu-opiate receptor, showing that, or indicating at least, that there was a high genetic risk. We also saw that when people came to the emergency room, even here at Mount Sinai, that the polymorphism of their mu-opiate receptor actually played into the severity of their drug overdose in their survival. So is it functional in the context that we could see in the human brain? And that's what we do in my group. So in studying the human postmortem brain, we could see that the polymorphism of this mu-opiate receptor, it's not just, it has a functional relevance to how the cell and the receptor functions. So it's downstream signaling, and that's so key. So it's not that people are just, their morals are bad, and that's why they become, a person that has an opiate use disorder. Their genetic impact, that even on how their cells signal to the downstream signaling cascades, to their transcription factors in their cells that make that difference. Behavioral traits and psychiatric comorbidity also play a role in this with genetics as well. And here I'm going to use the example of cannabis use disorder. We know that cannabis use disorder, about 30% of regular users do develop a substance use disorder. And we know that with cannabis use disorder, it has a high psychiatric comorbidity. For example, you can see, personality disorders, and you see a lot of anxiety disorders as well. And we know that a lot of the neurobiological changes that we see associated with cannabis use disorder actually mimics what we see for other substance use disorders. But such as, for example, that prefrontal cortical area, that orbital frontal area that's important for cognitive flexibility. We also see that there are significant changes in their amygdala. And the amygdala is really important in terms of emotional regulation and aspects of anxiety. And in our population, when we study people with a cannabis use disorder, we could actually see that they had really higher neuroticism, anxiety traits, and also aggression. Also, they had greater impairment of negative affect. So what about genetics? As I said before, many people will, well, I don't know if I studied this yet, but many people consume, cannabis, but they do not go on to have a cannabis use disorder. So why is it that we have a certain percentage that do. And when we looked at behavioral traits and genetic polymorphism, it was interesting. It doesn't matter the particular polymorphism here, dopamine D2 gene or proenkephalin opioid gene, but we could see one thing. If you had an anxiety neuroticism trait, it increased your risk just a little. If you had the genetic risk, it increased it nearly twofold. But if you had a combination of both, you had a greater risk of developing a cannabis use disorder. So we know that genetics does play an important role in who develops a substance use disorder, but it's much more complex because there are no single genetic factor that we can link to all substances of abuse and even to specific substances, but they all go together with aspects of the behavioral traits, and now I'll even talk about some aspects of the environment. So when we look at the environment, we know that early life events have important impact on adult behavior. And here, when you look at early life trauma, we see that even for substances such as marijuana, even cannabis, that the age of initiation is earlier. But it's not only for cannabis, it's also for cocaine and heroin use. So childhood trauma is a huge issue that we know is important for addiction risk. Karen Baki, an assistant professor here at Sinai, using neuroimaging and studying adults with cocaine use disorder, but who had a childhood trauma, we could see that these individuals have much greater impairment of their orbital frontal cortex than control subjects. And the greater the high level of trauma during childhood, the greater their depression as adults. So environment makes a difference. One aspect of environment that I've always found interesting actually links back to some of the key features that we start to see and people have replicated in different groups in terms of what are some common features of substance use disorders. And one of them comes back to the neurotransmitter dopamine and the dopamine receptor, this D2 receptor. And we know that in many adults with substance use disorder, whether it's psychostimulants or alcohol or even cannabis, we can see that there's a reduction of the dopamine D2. And this was observed by neuroimaging by Nora Volkov, who now heads the National Institute of Drug Abuse, and her colleagues way back in Brookhaven National Laboratories. And as I said, this has been replicated by a number of people. But the thing that's fascinating is that environment can impact on your dopamine D2 receptors. And I showed this very seminal work by Mike Nader and Morgan and colleagues, where they looked at monkeys. And here, these are individually housed monkeys. And when you socially house them, you can see that their dopamine D2 actually increases. So think about COVID, as I talked about earlier, social isolation. Your dopamine D2 is definitely here by these animal models reduced. But the thing that was interesting for me in their study is that not all animals, even socially housed, had an increase in their dopamine D2. Your social stress made a difference. These animals that had low D2, even when socially housed, were subordinate in their society. And these animals with that subordinate stress, they were the ones that self-administered more cocaine. So the brain neurochemistry really can give some insights about vulnerability and the aspect of stress. Now, stress also impacts on a big area for my research program on neurodevelopment. And for me, I'm going to focus this part about the endocannabinoid system, but mainly on cannabis. And the reason is that the endocannabinoid system plays a critical role during the neurodevelopment, from the fetal developmental period, actually even before the sperm and the egg. The endocannabinoid system is even critical there. But from the beginning of migration of, or making of cells, migration of cells, how synapses form, to later in life in terms of during adolescence and young adulthood, when there's synaptic pruning and the final maturation of the prefrontal cortex. The endocannabinoid system, our endogenous cannabinoid system, plays a key role. So the question is, what is the impact of cannabis during those time periods? And for me, I've called today in one way the perfect storm, because today we have wide availability of cannabis due to, the large legalization of cannabis, which I think impacted so much on a negative manner on especially Black and Brown communities. But it's also associated with very high THC concentrations today. And with now many people thinking that there is a very reduced perception of any risk for cannabis. And that to me is the perfect storm for the vulnerable developing brain. And we've studied, for example, prenatal exposure and looked at endocannabinoid system and a number of other transmitter systems. But I'll just tell you about one thing about the cannabinoid system in the adult brain before I go on to the prenatal brain. One thing is that the cannabinoid receptor, the primary cannabinoid receptor, the CB1 receptor in our brains, it's the most abundant G protein coupled receptor in our brain. It's not for smoking cannabis, as I always say. It regulates synaptic transmission. But during fetal development, and this is the human fetal brain, it's expressed in brain regions really relevant to what I've showed you earlier, like the nucleus accumbens, this ventral part of our striatum, the amygdala, really strong expression. Amygdala important for emotional regulation, and here the hippocampus, again, in terms of memory. So what do we see in terms of the endocannabinoid system and cannabis? So many research groups have shown the critical nature of the endocannabinoid system for actual hardwiring of the brain during development. So cannabinoid receptors and the endocannabinoid ligands help the pathways to form. We can see in the brains of fetuses whose mothers smoked cannabis that these microtubules that make up the cytoskeletal organization, they're impaired in the brains of these fetuses. However, really fascinating. Remember, I told you one of the key, common neurobiological marks of substance use disorders in adulthood is this reduction of dopamine D2 in the striatum. And we can see that during prenatal development in the human fetal brain, for example, their nucleus accumbens. We can also see that even in their amygdala. And here we can even see that there are sex differences, where the boys are much more vulnerable to their mother's cannabis use. And, when we study kids often with early exposure, we will say, oh, the boys show this greater behavior. And we say, oh, it's society. But here, this is in utero, and there's already a sex difference. So when you study the human brain, especially during neurodevelopment, the question is, in human studies, what's the specificity of the neurobiological findings that we have to actual cannabis? Because the moms will take perhaps other substances as well. But an important question for me was, what are the long-term consequences on the brain for adult, at least at that time, about adult brain and behavior? And so this is when we bring in our animal models. And our animal models actually replicated what we saw in the human fetuses. So for example, at the same prenatal developmental stages, that decrease in the dopamine D2, that vulnerable mark, was already there in the animals that have been exposed to THC. When we follow them into adulthood, that impairment of dopamine D2, that reduction, is still there. These adults have never seen THC other than when they were during the prenatal stage. We can even look at the electrophysiological properties of their cells, and they fire differently. We can even look at behaviors as adults. And here we're looking at, for example, are they more sensitive to opioids as adults in terms of looking at sensitivity to self-administer more heroin, for example. And we could see that indeed adults with prenatal THC exposure self-administered more heroin. And even under conditions where we kind of tweak the system so they would self-administer the same amount of heroin, they would run to the lever for that first heroin hit much faster, this latency. And whenever they're stressed, they would always take more heroin. So we knew back then that there is this long-term impact of THC in our animal models that we had initially seen from our prenatal human studies. But I always say, I was a little bit, not a little bit, I am masochistic. So I wanted to go back and look in the human subjects. And I also have an even more masochistic colleague, Yoko Nomura, who had been studying women with prenatal stress. And we wanted to look at also prenatal cannabis exposure and follow the kids throughout their lives. Obviously, we're not going to get to the end of their lives. They're going to hopefully, knock wood, outlive us. But we can begin to see certain things. But in order to try and get some indication about what was the microenvironment, that in utero environment that the kids were in, we decided to look at the placenta. And we sequenced the placenta to see what genes were changed, what's the environment on a gene expression level. And I'm not going to give you the whole things, but we could see, for example, that a number of genes were changed. And some of them are extremely fascinating, but I'm going to tell you mainly about one, about the cannabinoid receptor, for example, that's significantly reduced in the placenta of the moms that smoke cannabis. And it correlated to the amount of cannabis they used. So it shows that their endocannabinoid system is dysregulated, but so is their stress receptor system, their stress hormone system. So here, the glucocorticoid receptor in the kids that had both the double hit of cannabis and stress, they had a greater response. The same thing, double hit of cannabis and stress, they have a greater endocannabinoid dysfunction. What about their behavior? So like I said, we've been looking at these kids for a while. I'm only going to show a little bit, but clinically, we can see even from 6 months that the kids differed. And here I'm just showing like age 4. We see again this double hit that kids have greater anxiety, greater aggression behavior, particularly with the double hit. But if you just look even at the those with primarily only maternal cannabis use, you see that indeed their alterations of their stress hormone system, that you can see the cortisol levels are significantly different in those kids at 4 years old with maternal cannabis use during pregnancy. And we see, like I said, these differences on their behaviors of anxiety and aggression. And I'm not going to share it today, but we can see that a number of the placental genes of a particular system, immune system, that actually predicted their behavior later in life. So we can follow the kids yet into adulthood, but we can follow the animal models. And the animal models replicate a number of things, but they also show that in adulthood, these animals showed differences in motivation, in depression, and changes in their hedonic, in their mood state. So for example, for motivation, you ask an animal to press a lever to get 1 chocolate reward, because I love chocolate too. And then you ask them to press for 4, then you have to ask them to press for 8, and so on. These animals, adult animals with prenatal THC, will press a lever 100 times just for 1 chocolate. They also show, because that depression-like phenotype in a classic behavioral despair model, and changing of their hedonic state, their mood state. The question for us is, what are the neurobiological mechanisms that maintain these long-term effects into adulthood? We know, as I said, addiction is complex, and I showed you the, impact of genetics, but these are not genetic changes. Cannabis is not causing mutations. It's causing these epigenetic changes. So epigenetic changes, or epigenetics, is really describes mechanisms by which genes are turned on and off, not by changing your genetic code, but by changing, like I said, how, for example, genes that should be turned on are now turned off, and vice versa. I'm not going to go through, all of the mechanisms that we know, but we do know that there are certain molecular modifications that will lead to turning on a gene, so upregulating a gene, while other epigenetic mechanisms will turn down regulating a gene transcription. And when we look in in adult animals that had prenatal THC, we see a very interesting thing. We see that genes that relate to this epigenetic mark that is important for maintaining epigenetic memory, that it actually correlates really strongly, or not it correlates, that it regulates genes that are critical for synaptic plasticity. So my cursor is being very sensitive tonight. So from the data that I showed, and then there's a lot of data, I'm just giving you the overview. We know that prenatal cannabis, and especially from our animal models, THC specifically, that exposure can lead to these protracted disruptions in behavior that we can see in adulthood, on how the shape of the cells, the physiology of the cells, and importantly on these epigenetic mechanisms that are linked to changes in synaptic plasticity. So how neurons talk to each other. The next developmental window that, for us is really important is adolescence, because this is a time period when many people experiment with drugs themselves. And we've done a great job in our society of teaching kids and adults about, the health risk of smoking cigarettes. And again, I don't know why my cursor is being very sensitive, but so we've seen a decrease in cigarette use in teens, but cannabis use has definitely remained, has gone up over the decades. And with the cannabis use disorder, with the increase in THC, that becomes a huge problem. So we know there are a lot of long-term effects of adolescent cannabis use from human epidemiological studies, where the greater frequency of cannabis use is associated with, the greater illicit drug use later in life, or cannabis use, and a lot of, for example, suicidal attempts. And the earlier kids start using cannabis, is a big problem as well. But there are a lot of debates about that. And so the question for us, can we use animal models to start giving us some more direct causal insights? And when we look at our animal models, so we give adolescent THC exposure and study them, for example, in adulthood, and have them freely self-administer heroin. And invariably, the majority will self-administer more heroin. There are definitely behavioral traits on the degree of that, just like in humans, which is interesting, and research that we're also following up on. But given the fact that as I mentioned in the beginning, that the endocannabinoid system is really important for that final maturation of the prefrontal cortex that doesn't reach full adult maturity until even about mid-20s, we wanted to look at the prefrontal cortex and look at those neurons there. And one of the things that we could see is that when you look at adults that had adolescent THC compared to controls, there was not really much difference in like the length of their trees or of these cortical neurons. But what differed was the complexity of their cortical, these pyramidal cells. So there was a reduction in their complexity. And here on the right, you can see these are actual neurons. These are schematics from actual neurons. And you can see that the THC neuron, in terms of adults that had adolescent THC, their cortical neuron is quite diminished, the complexity of their cortical neuron. But I wanted to know what is happening on a molecular level in those cells. So we actually picked up those cells with a laser and a microscope, and then we sequenced them to see what was different that might have accounted for these morphological changes. And it was really fascinating, shocking actually, that when you looked at the gene expression profile in adults with adolescent THC compared to normal developing brain during that time period, there's only about a 5% overlap in their transcriptional signature in those cortical cells, only 5%. And those changes occur predominantly in relation to the dendritic, the cell morphology, how the neurons are developing, but also in regard to epigenetic mechanisms. Really profound changes in the epigenetic signature in these cells. And when you look at how these two systems, these epigenetic modifications and the regulation of these neurons, how they talk to each other, in normal animals, in vehicle-treated animals, there's not a huge overlap. But if you look at the adults with adolescent THC exposure, there's a complete reorganization of how these genes talk with each other. You don't have to be a computational biologist to see this. Of course, we have computational biologists to give us all the statistics, but it's quite clear that their cortical gene expression profile is quite different. So we can see both structural developmental changes in the prefrontal cortex, and we can see a reorganization of the gene expression network. And that's linked in large part to this epigenetic dysregulation, and also, again, to aspects of synaptic plasticity. So there is this long-term impact on the brain. It's mediated through these epigenetic processes, as I said, that we see both in terms of from heroin use and also from cannabis use. And again, two things that they share in common are these changes in synaptic plasticity. So a big question then comes back to, can we start using some of the insights that we're gaining from the brain studies to develop a way out of addiction, to develop treatments? So I'm going to use opioid use disorder in giving you some insights that have been leading us into new ways of treatments. As I mentioned before, opioid use, activation of the mu opioid receptor that we know that has a genetic aspect to the molecular disturbances that we see relevant to synaptic plasticity that leads to this opioid use disorder, or at least contributes to opioid use disorder. And when you look at the transcriptional profile, meaning when you look at the gene expression pattern in the brains of people who died, who are heroin users, we can see that there, significant dysregulation in those individuals. And the neuronal systems that are changed come back to dysregulation of glutamatergic genes. And this was studied in the striatum, for example. And when you look at this, we also saw that there were significant dysregulation of these epigenetic marks. So again, epigenetics and glutamatergic dysregulation. Importantly, when you look at, there's this huge correlation between these epigenetic marks and these glutamatergic dysregulation, primarily in the heroin abusers, not in control. And these epigenetic dysregulations, they actually correlate to the years of heroin use. So why is this interesting? This is interesting because we can start to see what type of epigenetic changes are occurring in the brains of heroin users. It's not every epigenetic modifications that are changed. In fact, not at all. They're very specific, even with cannabis as well. What we see are these changes in a particular epigenetic system dealing with acetylation to open up these glutamatergic genes. And we can actually replicate it in the animals that self-administer heroin. So we know it's heroin and not other things that the person's life might have had. Importantly, there are medications that are developed in the cancer field for epigenetics, because the cancer field is I call it disorder of epigenetics gone awry. And these inhibitors we use in our animal models. And if you infuse it in the brain, the striatum, you can reduce heroin self-administration. But of course, we're not going to have that as a treatment. But when we give it systemically, we can also decrease heroin self-administration in animals and also heroin seeking behavior in these animal models. So this now becomes a strategy for us in targeting epigenetic mechanisms as a way of normalizing perhaps or reducing heroin seeking behavior, where we know that the history of heroin use in humans actually correlate to the impairments that we see in the brains in relation to epigenetic disturbances and these glutamatergic genes critical for synaptic plasticity. Another way out in terms of strategies to what we've learned about the brain comes back to our research looking at the developmental impact of cannabis. So I started looking at the developmental impact of cannabis because we had seen often in people that had a heroin use disorder, or in people who had died from heroin overdose, at least in our postmortem brain collection, that often the information was that they had, used cannabis earlier. And of course, there are other drugs as well. And then our animal model actually helped to prove that indeed, early exposure to THC, as I showed you before, they would self-administer more heroin. But a thing that we had a bias when we are doing our animal models is that we, of course, want to have clarity as to what is really causing the changes in the brain. Because many people with a substance use disorder will take many drugs. They are polysubstance use, even if they may have a primary drug of choice. And one of the things about cannabis, when we were always, when we're carrying out our animal work, we say we're studying cannabis, but we're actually studying THC, because that's the cannabinoid that leads to the high, the rewarding effects of the drug. That's why people take it recreationally. And that's the cannabinoid that has gone up the concentration over the years. Other cannabinoids, there are over 140 cannabinoids in the cannabis plant, and actually over other chemicals as well. And these cannabinoids can have also biological activity relevant to the brain function. And one of them, we said, at least for me, I wanted to look at cannabidiol. Cannabidiol used to be much greater in concentration in the cannabis used on the street today. And so years ago, when nobody knew about cannabidiol, we said, let's take a look at another cannabinoid, since in our animal models, we were always saying cannabis, but really we were studying THC. And when we did that, it was very surprising. When we gave CBD, it actually reduced their heroin-seeking behavior. So when animals, just like humans, when they take drugs, the environment starts to take on important context. So for example, an animal is self-administering the drug, a light might go on in their environment. And all you have to do is to show them the light later, and they'll press the lever as if they're to try to get heroin, but they're not getting heroin. And that cue is enough to, as I said, to trigger, we call it drug-seeking behavior. And CBD reduced that cue-induced heroin-seeking behavior. And an interesting thing from our animal model was that even weeks after the final CBD administration, it still reduced heroin-seeking behavior. So although no one was studying it at the time, I had two choices: trying to figure out how CBD, the neurobiological effects of CBD, we're still trying to understand its mechanism of action, or to see whether or not this might be useful as a potential way out, as a potential treatment. And one reason why I thought this, not only that our animal models show, as I said, showed the behavior, but as I showed you, there's a lot of glutamatergic and synaptic plasticity dysregulation in the brains of human heroin users. And we can replicate those glutamatergic disturbances in our animal models, as I showed you before. But interestingly, when we gave CBD, it normalized some of those glutamatergic and synaptic plasticity dysregulation. So for me, the question was, could cannabidiol be used as a potential treatment for opioid relapse? And so we started to carry out double-blinded, placebo-controlled studies. And for me, it's important to have double-blinded placebo-controlled studies, especially for cannabinoids. Well, for THC people, it's tough because people always know if they're getting a THC. But for CBD, since it doesn't have rewarding properties, it's easy to carry out placebo studies. And what we saw was in people who were heroin abstinent and they were shown video cues, those people who were shown later to have gotten placebo and they saw a heroin cue, they craved, and CBD reduced that. And just like our animal model, when we brought them back in a week later, CBD was still reducing their craving. one of the things that we hadn't studied at that time, but now we have in our animal model, was the fact that CBD was having an impact on anxiety. So when people had been shown the video, the heroin video, and had received placebo, there was increased anxiety, and CBD reduced that. Once again, a week later, CBD was still having an effect on reducing their anxiety, their cue-induced anxiety. These are all self-report. So we also looked at physiological measures like cortisol. And when placebo subjects had been shown the drug cue, their cortisol levels went up, and CBD reduced that. Similarly, for their heart rate, they saw a drug cue, their heart rate went up, and CBD reduced that. So for me, based on our work, we think that cannabidiol does hold promise for opiate use disorder treatment, at least for the craving and anxiety. And of course, we still have to develop effective dosing and treatment regimen and the formulations or the delivery systems that may be most appropriate for developing medicines. And so that's why we're in the process of trying to conduct large clinical trials. And we're also looking at doing neuroimaging to get back to understanding what's the neurobiological processes by which CBD is impacting on these behaviors. Since I started, CBD now is everywhere. It's in our coffee, it's in our ice cream, it's in our skincare. I hope it will work, even in hamburgers. And wherever I go, everybody always tells me that CBD helps their dogs. So, but I hope that we get back to medicine, because it's really critical that we figure out whether or not this particular cannabinoid might indeed be a way out of the addiction that we see, and that not just CBD, but perhaps these targeting these epigenetic mechanisms that are quite specific in their dysregulation that we see with substance use disorders may be a way out for us to give people their lives back who have a substance use disorder. And with that, it's an amazing team of people, present and past, in my group, who have done amazing work. I mentioned Yoko Nomura, who is my partner in masochism for these long-term longitudinal studies in kids, and Ed Salsis, who is an amazing addiction medicine doc, amazing with his patients. And a lot of the people, Jackie Ferlin and Issa Barr and Greg Rumpala and Alex now doing all the developmental cannabis work, and from our human studies, and Li Wang and Karen Baki, who are heading a lot of our neuroimaging and the clinical trials, and this is our clinical team. So thank you all, and I will take any questions. Let's hope I didn't go too far over. Thank you.
Thank you so much, Yasmin. What a wonderful talk. And we have a lot of questions, as you can imagine. And I'll remind people, they can raise their hands going down to the reactions pane at the bottom right of their Zoom, or they can send them directly. Yeah. And we have quite a few sent in, and we have some hands. So I'm going to start at the top here. So did someone want to know, did you mention smoking, did tobacco come up in your talk?
No, tobacco didn't come up in my talk. I'm very biased. Yes. Clearly most of our patients who have a substance use disorder as opioids or cannabis, they will also smoke cigarettes. And we normalize for that on a statistical way to see whether or not nicotine may be contributing to some of the effects that we see. And definitely we see effects that are more specific for nicotine as compared, for example, to cannabis. But that's why, for me, it's really critical to do these animal studies as well, because there we know what the animal's history, and we can be clear about, this is heroin induced or this is THC induced. But nicotine is an important substance of use, yes.
Thank you. Okay, we have two people ask this question, our regular participant Nirav and also Christina, and I'm sure you get this question a lot. What's your opinion of Dr. Carl Hart's recent book, Drug Use for Grown-Ups? How accurate is it that the majority of people who try drugs do not get addicted?
Um, I haven't read his book, so I don't want to give, like, I know Carl, I haven't spoken to him in a while, but I haven't read this book. And as I mentioned earlier, that's why I went through the whole thing of, what contributes to the complex aspects of addiction, because a number of people will experiment with drugs, but only a certain percentage go in it. And I mentioned like genetics, early environmental uh, exposures to stresses, and even the drug themselves. But early drug exposure absolutely makes a big difference. Genetics makes a big difference. Um, there are so many things that contributes to why only a certain percentage of people go on to have that. The question to me is, thank God, that it's not a higher number. But if you look at the numbers, it's still pretty high. And it's critical that these individuals are given opportunities, as I said, for treatment. And the earlier we can identify people at risk, and not have them go deep into their addiction, the easier, not easier, but the better it will be to have a better outcome for their treatment. So yes, he is correct. And yes, there's a lot of stigma with addiction that has placed, as I said, I actually don't believe, I used to believe that, people should be locked up for addiction, because I was, you're raised in that way. And for me, I think that's ridiculous. I think that we need to have a different system. It shouldn't be within the judicial criminal system. It should really be about, it's a healthcare problem. And funds should be moved from the criminal justice system to a healthcare system to improve the outcome. If we really think that this is a disorder, and it's absolutely a brain disorder, it is absolutely a brain disorder. And many people are lucky that they do not develop a pathology that I'm sorry, I'm going off, but we still have an opioid epidemic. People are dying. So when people tell me that, oh, it's only a small percentage, or it's not really a disorder of this or disorder of that, I'm sorry. We see the significance every day. At Mount Sinai, we have one of the largest opioid treatment programs in the country. This is real. So, patients and their families and communities are highly impacted by it.
Yes, very important point. I'm going to ask Louis Schwartz to unmute and ask his question.
Thank you. I have actually two questions. From a layman's point of view, your description of cannabinoid use as far as making it more likely or making it easier to move on to other drugs seems to be the argument that people have made against decriminalizing marijuana use. It seems to me that your research seems to be supporting the argument that cannabis use is dangerous, not because it's inherently dangerous, but to use the overused term, it's the gateway drug to more serious addiction and more serious drug use. So could you comment on that? And the second question I have, when you talked about CBD being in everything now, what is the effect of CBD being in everything in terms of affecting the population at large?
Thank you for your questions, Louis. So in terms of the gateway, definitely our studies show that developmental exposure to THC does have a long-term impact and can increase sensitivity to opioids. However, as I try to emphasize, other things go into it. If you are a rat with a bored life, you probably, you come into a room where you're given heroin and no other choice, you're going to choose probably heroin, right? If you're animal studies actually show that if you give animals actually a choice, they will choose the other like a social outcome rather than a drug outcome when they press that lever. So I do think that the animal research is important, hands down, because it helps us to gain insights into the neurobiology. And hopefully the neurobiology, we can, as I said, that's what I use to then try to develop medications. However, it's not reflecting the real world situation. So I do want scientists to be involved and physicians to be involved in policymaking in terms of how we go about helping people with substance use disorder. I don't think that it should be criminalized, but I don't think that people should make it seem that cannabis is, is benign. No one would be smoking cannabis if it didn't make them feel a certain way. It does change your brain. The question is, do we need to have THC concentrations that are, in the 1970s, 80s was 4%, and now it's going to be up to over 20%. And with the dabs are 80% THC, that's a different drug. That has a huge impact on the brain, and especially the developing brain. So I don't think it's an either or. It's a complex situation. It's a complex problem. And for me, the animal models tell you the potential. And even our animal models, we see that behavioral traits make a difference even in our animal models. So if you look at the environment, so why don't we go in and help people earlier? If you look at the stressful lives in certain communities, if we tell certain communities that cannabis is okay, but they're most at risk, that's also dangerous. So for me, it's about education and using all the data from science, from, every single thing to be able to make those decisions. In terms of your second question about CBD being everywhere, CBD concentrations that are in that hamburger or in that coffee are not the concentrations that we're using clinically. So they're very low. And because so far, the data does not show that CBD has, especially at those concentrations, have any adverse effects. I mean, for me, you're just paying a lot of money for that coffee, for that CBD water. So I'm not so worried. I'm more worried about where the CBD comes from in terms of it being adulterated with other things. It can even have THC. They have found mold and all these things. So it's very important that people are wary about the CBD that they use, because there are a lot of companies out there trying to make money off this CBD, range, and there are a lot of unscrupulous companies.
Thank you. Okay, we have, I've consolidated these two questions, they're related. Melinda and James, the talk is about SUD. There are non-chemical addictions like digital, gambling, or sex. How similar are these addictions to substances and how the brain is affected or involved? And do these types of addictions also have epigenetic effects, is the other question.
No, I mean, that's a really important question. Obviously, I'm biased because that's what I study. I studied chemical addictions, and the chemicals impact the brain in those neurobiological systems, even the dopaminergic system, in a very profound way, in a much stronger way than some of the behaviors, the behavioral addiction. So, cocaine, it's elevating dopamine to extreme levels. Gambling will never give that degree of dopamine increase in the brain, for example. But the behavioral addictions do work on those dopaminergic, those mesocortical limbic reward system, and change glutamatergic systems also in the brain. The degree may not be as strong, but you don't necessarily need, perhaps, I should say this, we don't know what specific structural and molecular changes are needed on the behavioral level to give you similar things and these long-term effects as you have for the actual chemicals. But we know that they have these long-term impacts. So we know that there are epigenetic changes as well. They're animals that will run forever, like certain humans. That's not me. I'm not going to be running at these exercise. I don't get that endorphin high, but there are people and animals who do. And you can see these molecular changes, some of them very similar to the chemicals. So I think that we can learn a lot. And, even with neuroimaging, you see food addiction changing the dopaminergic system, the classic, changes, as you see with substance chemical abuse as well. So there are definitely a lot of shared neurobiological systems.
Okay. Next question, I'm going to ask Jacqueline to unmute herself. She's there.
Hello. So I have a question regarding the decrease in complexity in the prefrontal cortex, as you previously mentioned. And I was wondering if your studies have looked at any cognitive tests or any changes that might have resulted from those, from the decrease in complexity, and how that might have affected the neuronal structures.
Yeah, so it's interesting. We tried it for that study to look at a few behaviors, and we didn't see any behaviors directly correlated to, I shouldn't say correlate because there were some different animals, but we have now been able to look at different aspects of cognitive flexibility, but we have not correlated that to the morphological changes. So I don't know if they're correlated to that, but we see, the orbital frontal and prefrontal cortical region, for example, that plays a role in that cognitive flexibility, and with, aspects of, well, not just only cannabis, but even opioids as well. So I can't say that what specific cognitive effects that those had. However, the effects were pretty profound.
Okay, we'll take another of these that wrote in. Amanda has actually two questions. Did they find polymorphisms through GWAS or some other method? And how does the field determine cannabis use disorder? For instance, using the drug to treat anxiety versus having comorbid disorders. Is this just a difference in terminology?
No, so I mentioned earlier, the diagnosis is the DSM, the diagnostic manual that is used. So cannabis use disorder, many people, for example, think, oh, you can't become addicted, quote unquote, but there's a cannabis use disorder, and it's meeting, you either have a mild, moderate, or severe cannabis use disorder, just like it is for any other drug. So it's looking at those 11 criteria. And if the person takes the drug, despite trying to reduce the amount, whether it disrupts their lives in certain ways that have negative consequences. So just like other substances, so that's what's used to make that determination of cannabis use disorder. Wait, I think I forgot the first part. It was. What does it? Oh, sorry, Dorian, I made you have to. Wait, it's muted. Oh, wait, it's muted.
Did they find polymorphisms?
Oh, the GWAS. Yeah, so our studies were not GWAS studies. So because we have a very small sample size, so we looked at specific, based on the biological candidate. There are GWAS studies that have been done for cannabis use disorder, for most substance use disorders. So I will tell you, for people who don't know, when you look at genetics, and with and you're looking across the genome, you can say in a very unbiased manner, for substance use disorder, for a number of disorders, you need to have this magical statistical power. So but do you have statistical power? Do you have enough people to make that determination that it meets that genome-wide level for significance? And substance use disorders is very challenging. And I think that it's challenging. You need thousands and thousands of people. And it's challenging for a couple of reasons. One reason, for example, when we talk about substance use disorders, you can have the genetic risk, you can have the environmental risk, all of the risks that I talked about. But if you never take the drug, you will not get the disorder. So people who might have the genetic risk, but never take the drug, they will end up in a control group. And then the opposite, you can have someone who doesn't have the genetic risk, but they're taking repeatedly a very potent drug for their brain. So like heroin or the opioids, the oxycodones and so on, repeatedly, that can put them in that substance use group. even though they don't have the genetic risk. So that's why you need thousands and thousands of people to start getting a signal as to what may be that true genetic risk on that 1,000 level platform. And so that's one of the things that is still challenging, I think, for the substance use field.
I'm going to put in the next question here from Scott because it's related. Would it make sense going forward for pain management doctors to test for the DNA opioid receptor polymorphism in determining whether it might be safe for some patients to be treated for pain using opioids if they do not have the polymorphism and are thus at lower risk of addiction?
I think so. And I mean, a number of people think so as well. But coming back to the question before is that, it's not, so for opioid use disorder, they have now done a GWAS, and actually the mu-opioid receptor did turn out to be, for that, some of those studies, to be one of the candidates for vulnerability. So they've done studies where people, when they're in a hospital and they're pumping for their opioid, that it even, in fact, match onto their mu-opioid receptor polymorphism. So I do think that that's something, as we get to know more, and genetics can help pain management. Absolutely. And even, to be honest, at Yale, they're even looking at aspects of methadone maintenance. What's the dosage? Because everything is, unfortunately, there's no exact anything. And so you play with the dose that matches to a particular individual. So even that might be, genetics might be very helpful even in opioid maintained dosing.
Next up, we're going to ask Annette to unmute herself. Annette, are you there?
Hi, yes. Firstly, thank you so much, Dr. Hurd, for that talk. It was fantastic. And I think that you pretty much answered my question just now. I wanted to kind of touch on how your work can be translated into a clinical sense so that we can tailor our therapies. So I'm curious, do you think that would also be effective with treating opioid disorder, or figuring out who would best be suited to use medical marijuana instead of opioids for pain management?
I do think that that's where, where the field is going. The more that we can learn about genetics and who's at risk, I think that that's definitely something that everyone wants to be able to bring more into the substance use field, into psychiatry field. We don't have the EKGs, we don't have the, what's your blood pressure level. And so I think that that's one thing that will be very helpful. But I think on a translational level, I mean, clinically, addiction doesn't, and Saul says he always, that when I first met him, it's like, addiction, unlike many disorders, there's not a staging of addiction. The syndemic of addiction, we don't consider all of the different factors of the psychosocial factors. So genetics, yes, is one thing, but to me, I think you have to ask patients all of these different things to really get a better sense of what that person should be treated with, and what was their family like, what's their parents, issues. Just like any other disorder, if you know that your uncle had this and your aunt had that, alcohol use problem, you don't even need a cheek swab. There are a lot of things that can already tell you that you have a greater vulnerability, or you may have greater vulnerability.
Melinda has a question. Besides treatment for addiction, what can a user do to repair the damage he or she has done to their brain? I think how stroke victims get rehab and therapies to reprogram their brains. What about addicts who are sober and in recovery and no longer using?
Well, sober, in recovery, and no longer using is critical for healing the brain. One of the things I didn't emphasize, and obviously this audience, it's like the degree to which I'm not going to go into the detailed mechanisms of epigenetics and so on, but a critical thing about epigenetics is that it is reversible. So I'm actually hopeful, based on the molecular changes that we see associated with substance use disorder. It's not genetic. So yeah, if we take away that smaller percentage that have a genetic risk, and they will definitely be, much more vulnerable. The majority of people, the epigenetic changes that these drugs induce, they are reversible. They're not mutations. So we know that, for example, behavior can actually reverse or can lead to epigenetic changes. That's one of the things that we do have to work on, is to let people know that the molecular changes going on, it's not that they are damned for life, they are not. It's just that we have not done a really great job of impacting on every level for that person. So yeah, in recovery, not taking the drug. What does that mean? Are you filling your life with something meaningful? Are you having, the relationships that people have? Again, that big psychosocial component to their lives is really critical for positivity and helping to reverse some of these epigenetic changes. So I do think that recovery is possible. I don't think that perhaps we still have a structured strategy in the substance use field for dealing to like have a playbook that everyone can just check off and have as a prescription. But I hope one day that we can.
Okay, I'm going to ask Alex to lower his or her hand. And sorry for the wait there, Alex. Okay.
Oops.
Lost Alex. How about we'll ask Mela.
Oh, Alex came back.
Oh, Alex came back? Alex came back. Alex, you can unmute and ask your question.
Okay.
All right. Well, we'll try Mela then. Mela?
Hello, thank you so much. Can you hear me?
Yes.
So I've been fascinated when I was seeing the CBD combined with a lot of these different systems, wondering if it is, I guess I have a two-part question, wondering if it is actually acting as though it's an epigenetic kind of switch for these types of pain management. I'm currently doing a fellowship in palliative care, and it's fascinating to see if we're going to be able to, or if you're aware of, I guess the second question would be, are there any studies that you're aware of combining CBD with opioid use disorder patients in conjunction with some of their pain management issues?
So there are definitely studies going on with CBD and pain management. Earlier studies that looked at pain with CBD, it was never CBD alone. It was often with Savatex, which is a one-to-one combination of CBD and THC. So we don't really know if CBD is going to be great for pain management without some THC. And so those are why, those CBD only studies are critical. And even to have an opioid sparing. So you might not need to get rid of all the opioids. Opioids actually are critical clinically. It was the chronic use of high dose opioids that caused the problem. So if you can reduce the amount of opioids that's needed with CBD, that may be great already. So that's why some of the studies are doing that. And your question about CBD and epigenetic switch is exactly what, some of the things that we think is happening. And I can't go into some of the results, but definitely there are aspects of CBD that's fascinating. And CBD, I always say, has made me think differently about how we develop medications. So normally pharmacology, we're taught that you develop the very selective, like agonist to, knock the brain, that particular receptor, or a blocker, it's very selective. But all of those strategies have led to these bad side effects. And CBD is interesting in that its mechanism of action is, well, we're still figuring out many things, but it's actually not an agonist at the cannabinoid, the CB1 or 2 receptor. In fact, it's even an inverse agonist, having a little antagonistic effect. But what it does is that it impacts a number of transmitter systems in just a little way. So it tweaks many systems, especially related to the synapse. And I think that may contribute to why it doesn't have these bad side effects, because it's not this big hammer on the brain on all those mechanisms. And that, I think, is something that has made me really rethink about how we go about developing medicine.
I think you also answered someone else's question about whether CBD might reduce nicotine craving. It sounds like
Yeah,
needs to be
there are
studied.
some studies that are at least for like, the stimulus properties that nicotine has. There's been some studies showing that it reduces that. So it will be whether or not now it actually reduces intake.
Okay. Esther has a question. You talked about the developmental effects when younger people use THC. How do the findings evolve, if at all, when looking at adults 30 years plus beginning using it? And thank you very much for the talk, she also said.
Thank you. So my research has not looked completely in the adult versus, we definitely, your brain has a drug, even as an adult, you're going to get certain changes. But people who have looked at those studies, it is not, you don't see the same degree of change with drug exposure, not just THC, drug exposure during adolescence as you in adulthood as you do in adolescence. There's something about the developing brain that is much, much more sensitive. We don't know why still. Obviously, it could be, as I mentioned, that aspects of the endocannabinoid system and a number of other systems that are still developing well into the mid-20s. And that's what makes that vulnerability. But it's clear that the adult brain, you're not getting acute psychosis in your mid-30s as you do as an adolescent with cannabis. You don't see that kind of association. So definitely the adolescent brain is much more sensitive.
Okay. I'm going to give Alex another try here. Alex has shown up. Can you unmute yourself, Alex?
Yes, can you hear me?
Yes, we hear you now.
Sorry, I was pressing the wrong button. Could you talk about byproducts of combustion when one is smoking pretty much anything, namely carbon monoxide and hydrogen monoxide? And also, second question, on your list of addictions, you mentioned addiction to sex. Is there a treatment for it?
So I'll take the last one. I mean, I didn't mention anything really related to sex. We don't study behavioral addictions, but I mean, in terms of sex addiction, I don't know of any chemical treatments, but there are behavioral treatments that definitely have a lot of people have used for people who have a sex addiction. In terms of like the combustion, hands down, we talk about smoking for nicotine and for cannabis. One of the things in terms of like for, even when people are thinking about cannabis as medicine, people will say, oh, but cannabis by itself, but nothing that you smoke is going to be medicine because you're bringing something and the combustion, like you said, into your lung. And we know what even vaping had done in causing a lot of lung damage in young people, for example. So, it's not just the lung damage, but these things also impact on the brain. So absolutely, the combustion, those byproducts are really critical in terms of being the negative one of the negative aspects of smoking anything.
Okay. We have a question from Anasuya who says, are the reduced dopamine levels visible in those predisposed to addiction also associated with psychiatric conditions like ADHD? Do we have evidence to suggest how early diagnosis and treatment of such conditions might influence the development of substance abuse disorders?
So the dopaminergic system absolutely in ADHD is, a classic classically found there. And, that's why they give a lot of the kids these psychostimulants to increase their dopamine levels and so on. We all know that these development, for example, like I showed you that this high anxiety, high aggression, and actually ADHD is a risk for substance use disorder or psychiatric disorders later in life. So it is critical, and many people, it's funny, we think of psychiatric disorders as adult disorders, and they are in large part. However, we all know now that most psychiatric disorders, I would say, are developmental in nature. We are just now trying to understand the developing brain. We have to go much further back than we thought in trying to understand psychiatric disorders, because they absolutely start early. And that's why we do need to get much better understanding of the developing brain and what are interventions. It doesn't have to be chemical interventions early, but I think behavioral interventions are really critical to have early on.
Okay, I'm going to ask Ashley to unmute. Ashley?
Hi, Dr. Hurd.
Hi, Ashley.
So I have a question. Let's say for a teenager who has been suffering from drug addiction, would you say that it is easier for them to recover from it compared to an adult?
You know, I don't have empirical data to say in terms of if they will recover quote unquote better, except because it depends on when even the adults started their substance use disorder. But if we say that someone starts, as I mentioned, the earlier someone starts, the more at risk they are of developing, worse problems later in life. But the adolescent brain is still developing. The plasticity is there. So this is a time, that's why we said, this is a time for intervention. And so it's not that the adolescent brain, that it's harder. In some ways, because of that great flexibility, that great plasticity, it may be easier to intervene earlier. So I absolutely think that we wait too long for treatments to be initiated. And when you then wait when somebody's well into their mid-30s after, abusing for 20 years, that's very different than intervening when somebody's 2 years into their disorder as a teen. So absolutely, the teenage brain is very plastic, and we need to spend more effort during that developmental window.
A question from Clara, who says, does methadone maintenance that mothers get for their children prime the children for addiction later in life? Also, does the use of Adderall and methylphenidate among pre-adolescents do the same thing?
So, a lot of people are always thinking that, pregnant moms who are on methadone, they should stop. However, the data shows it's much better for the moms to be maintained on their, whether buprenorphine, maybe even better, methadone while pregnant. Again, I want to emphasize there are many things that go into why somebody may be more vulnerable. So yes, if you are exposed to opioids prenatally, that may set you up for greater sensitivity. But the interventions that come in, the environment that is there postnatally is also critical. So that's why we have to make sure that the women get the support they need, not just, both prenatally and postnatally, because that's what's going to be critical for not having these, again, these epigenetic changes are reversible. The earlier you get in there, the better. So for me, absolutely. And in regard to, treating teenagers with, Ritalin, methylphenidate, and so on, the data, initially people thought, oh, all of these kids are going to become more addicted, and that didn't happen. So there's so many, the studies are actually clear. So I would still say that the kids who have that ADHD should be, well, talk to your clinicians, but I think that the data doesn't show that they become addicted more.
Dr. Hurd, I feel like I should ask you now how you're holding up with all the questions.
Oh, I'm good. It's great questions. Yeah.
I need to go. We're going to ask from Michael.
Great. Thanks. Fantastic lecture, Dr. Hurd. Could you contrast, say, the effect of opiates and cannabinoids with the effects of alcohol on the brain, and perhaps epigenetic effects that alcohol addiction would have on plasticity and epigenetic factors?
A lot of the things that you see in the brain with, cannabinoids and opioids. You do see with alcohol. I mean, alcohol is a much a broader effect in one way because it's a depressant on the brain, and depending obviously on your alcohol use disorder, but you see many of the neural systems impacted with alcohol. One of the things I can say is that animal studies have shown, they replicated what we saw with opioids, that CBD even decreased alcohol use, and even months after their final CBD administration. So, a lot of the changes in synaptic plasticity and the epigenetic changes that people also see with alcohol use, these long-term effects, there are common mechanisms that we can indeed target for cutting across different classes of drugs. And I actually think that that's important. They're very, very rare do you find someone who never takes any other drug, only have one drug. And so I do think it's being able to give people certain things that may help them achieve abstinence, not just from one drug, but from a number of drugs, by the fact that we target more common mechanisms in all of these substances.
Okay. Shifa would like to know if hypnosis can work in some extreme cases of addiction.
I'm not knowledgeable on hypnosis at all, even though I would love to get hypnosis for some of my bad habits and help me with sleep. I do think that there are people who have sworn by hypnosis, and I think if something works for you, and that's great. So absolutely, but I don't know enough to really give that much insights about the neurobiological effects of hypnosis. I know meditation can change certain parts of the brain that, calms parts of the brain. So I'm sure hypnosis can do similar things.
Okay, we're going to ask Elevation to unmute.
So Dr. Hurd, what was the racial and gender makeup of the double-blind study of the 42 recovering heroin addicts that you conducted? And the second question, do people with the Neanderthal gene suffer from opioid abuse more than others?
The Neanderthal?
The Neanderthal gene, correct.
Okay, I'm not too sure I understand that completely. Probably somebody can help me, but I'll answer the first part. So in terms of sex, we had more males than females. And that's one of the things. So for our clinical trial that we are conducting, that we're about to embark on, that's one of the things, is to increase the number of females. So, we need to be able to see whether or not it will be equally effective. I can say that in animal models, there is a sex difference that people have begun to see, that females need a higher concentration of CBD, a higher dose of CBD, to get the same decrease in drug seeking or drug taking. So we'll see if that also replicates in humans. But in terms of the gene, I'm sorry, but I don't know why I'm like, in terms of what gene you're meaning, what is the biological specificity of that gene?
Yeah, so Caucasians tend to have the Neanderthal gene more than other people.
Oh,
maybe there's a different name for it.
No. Okay, now I understand what you're talking about on an evolution level. Okay, so I mean, absolutely, there are differences in between races in regard to certain susceptibility to certain drugs. So, for example, opioids, we see that for like the polymorphism of the mu-opioid receptor that I mentioned, Asians have a much greater frequency of that polymorphism. And when you look across the globe, it comes back to like where people were. I mean, Asia, a lot of poppy plants. There are reasons for why we may develop these evolutionary genetic traits depending on our environment. So yes, for alcohol, there are certain Caucasian communities, genetics that have a greater risk for alcohol use disorder. So absolutely, aspects of genetics can play with race, but there are a lot of social factors that also play in, that I don't think that we have, a great, and especially for African American or African in general, Blacks on the planet, we don't have great genetic data at all, since most of the studies have been done on European American populations or European white populations. So, there are big studies, more studies taking place in African ancestry communities. And I think that will help us a lot.
This sounds like it needs to be de-skewed a little bit there.
Yeah.
Paul has a question. Is premature birth considered a childhood trauma that may play a role in addiction in later life?
So, I mean, it comes back to, premature in regard to, oxygen in terms of circulation, in terms of brain. So, I mean, some people think so as well. But again, I want to keep emphasizing it's not deterministic. So many things, go into what really, who will develop a substance use disorder, because as we emphasize, the majority of people will not develop a substance use disorder, and which is a great thing. But the fact is that we still have millions of people with a substance use disorder. So I don't want to ignore that at all.
We're going to ask James to unmute. James P.
Yes, Dr. Hurd, I had a quick question about the over-the-counter CBD that you can purchase. Is there anything out there that comes close to the concentrations that you use in your study, with CBD and its benefits in alcohol use disorder and opiate use disorder?
So I will be very honest. The doses that I used in humans were actually based on our rat study, because back then when we started, nobody was really studying it. So I really didn't know where to turn. And those doses are nowhere near what's in, the over-the-counter. So we use 400 and 800 milligrams. In the over-the-counter is 10 and 20 milligrams, right? And what people don't realize also is a lot of these cannabinoids have really poor bioavailability in a lot of especially over-the-counter, obviously, preparations. So, you're not getting that much in your body. Do we need to have such a high concentration? We don't know. So that's why we're, doing research to try to look at different doses. And so I think that we're still pretty early in understanding what doses are really quote unquote clinical. Well, one, which subgroups of people might benefit. I do not believe that there is a one size fit all medication for anything. So I do not think that CBD is a cure all to any stretch of the imagination. We have to do a lot of research, and we have to study a lot of doses. And to the person who asked about the sex before, elevation, in the context of gender as well. So for me, the over-the-counter may work. And I do know that some of the over-the-counter have worked with people who I absolutely trust, in their, can there be placebo effects? That could be. And I'm actually fine with placebo, if it works for you. And they found that it, decreased their anxiety, and that's great. And so, but do we need, 400 milligrams for everybody to do that? Perhaps not.
Another CBD related question from Jimmy, who says, thank you for the talk. In your slide, you revealed that the structures of THC and CBD resemble each other a lot. Are we seeing evidence or the potential that CBD can have negative effects on our neurodevelopment?
Again, those CBD developmental studies a number of them are underway, so we'll find out soon. I want to emphasize, even though, the cannabinoids may look similar, they actually have very different actions in the brain. So as I mentioned, CBD is not an agonist like THC is at the cannabinoid receptor. When the agonist of the cannabinoid receptor is what induces that high, the reward. CBD does not have that. It's actually a poor agonist at the cannabinoid receptor. It is even an inverse agonist, as I mentioned, which is a kind of antagonist. So it's really important. They may look similar, but they are actually not in a lot of their mechanisms. Do they have some similarities, like both can, inhibit inflammation and all that? Yes, they have some similarities for different transmitter or neurochemical systems that they impact, but not on at least aspects of reward and addiction.
We have a hand up from Chris. Chris, can you unmute?
I think I'm unmuted. I'd like to come back to the CBD dosage. I believe I noticed in your great graphics that the larger dose you often gave, or in a lot of the cases you did do comparative dosages of 400 and 800.
Yeah.
And it seemed to me that in most of those, most of that data, that the 800 was actually more effective.
Is that just your question? Okay. Yeah.
That's
so,
the question.
Yeah. So it does appear that there's a dose effect, but it wasn't significant for many things. And it's probably not significant just because our sample size was small, relatively small. So that's why we do need to have these, this large clinical trial that, I've worked hard for years to try to get to be carried out. Then we'll see whether or not they're, the dose effects are significant. And as I said, some people might need the higher dose and others may not. And depending, on other comorbid aspects. But yeah, there tended to be a dose effect. But I would still go for the lower dose to start.
Aaliyah has a question. Considering the comorbidity of addiction and other disturbances in emotional regulation as seen similar in PTSD, depression, and anxiety, has there been consideration for future research with CBD and adult autism spectrum disorder symptom treatment?
Yeah, there are studies, and I think even in Israel, they're conducting studies with autism. So, CBD now has become this, panacea drug, which does scare me. As I said, I don't, I hate this, that people think that there's a cure-all, there's one cure-all. But, perhaps it's going to be about dosing. So for certain disorders or conditions, you might need this particular high-level dosing, and for others, you might need this. And so can it work with autism? We will see. But there are studies being conducted now with autism populations, both children and adults.
We have quite a few questions about cannabis and motherhood. Let's see, how much cannabis needs to be used by a mother to cause these changes in her unborn child's brain? Pregnant moms don't always know they're pregnant until they're 4 weeks along. Will cannabis use only during those first 4 weeks also affect the developing fetus's brain?
That's a great question, and it's one that is tough. So absolutely, early exposure can have an impact. However, in our studies with Yoko Nomura, for example, my postdoc Greg Ramapala, who's been doing a lot of these studies with the placenta, and we see that is actually more the later stages, mid to later stages of pregnancy, where there's a greater impact. So, I think that people, as soon as they know they're pregnant, should decrease the things that can impact on their fetuses. But it doesn't mean that what you've done early is going to be, again, deterministic for that person's life. I will keep emphasizing the changes that we're seeing are not mutations. They are not lesions in the brain. They're epigenetic. So they're reversible. Kids, the positive, the unconditional love, and positive environments that, the education that they receive, all the things makes a huge difference for, their outcomes. Does it mean that all of those things can prevent someone from having a psychiatric disorder? Probably not, but it's a combination of a lot of different things.
Okay, I think you just answered the next question from Roland, which was, are epigenetic changes caused by chemical substances reversible or are they permanent? Which you just now actually answered. Are you restricted from going into the results related to epigenetics because that would be a violation of NIH rules on proposal reviews? No.
No, no, not at all. I mean, do I push NIH for certain things? Yes. I really think that, but it's not NIH. I think people, well, some things I will say, but you're talking about colleagues, because you write grants and you have to convince your colleagues that the questions that you want to pursue and the manner in which you want to, the research design that you want to, how you want to conduct them, that they are will be carried out well, they are feasible, and that they're important. I will tell you, for many years, no one believed that my research in developmental effects of cannabis was important, or even cannabidiol was important. I got bashed. And, so sometimes scientists can get it wrong. So but yeah.
Okay. Christina wants to know, what do you think of the effect of puberty blockers on the brains of young people who are preparing to transition to a different sex?
Oh, that's so complicated. That's very complicated. I mean, clearly it's a whole organism change, a whole brain change. And it's not just the chemicals, again, that it's everything that comes with it, with their environment and the people and around them and supportive and all of that. But it's a huge chemical change on the brain, clearly, and on their body, but clearly.
One person did ask, how can we continue to keep in touch or contact Dr. Hurd even after the end of the discussion?
I am out there in the public because I am at an academic institution, so my email is readily available. So please reach out. Absolutely.
I think they can probably follow your research on there too and see what you're up to. What are your feelings on legalization of hard drugs, which has shown a significant reduction in drug use in a European country who did that, thus moving the criminal out of the distribution chain?
Yeah, as I think I emphasized that a little earlier, the country like Portugal, I think that's really critical. As I said, it has not worked, right? It has not worked to imprison people for substance use disorder. In fact, it has worsened the condition. So let's just try something new. Oregon actually is the experiment against the state in the US, because they voted last year to take away drug use from the criminal justice system. But I think that should be across the country. In psychiatry and addiction treatment in general, we just need to have we need to push a reset button and have a different way of how we treat these disorders. And once we do that, once we get rid of the stigma and all of that, we will decrease the numbers dramatically. And that's why I think that people don't realize.
Okay, John wants to know, if you were not resource constrained in any way, would the focus of your research change? And if so, in what way?
Wow, John, that's a great question. I would love if you could donate so that I would not need to keep begging for writing grants. And I truly believe that we can come up with treatments and novel way of treating. I didn't mention like one of our research where we, and again, looking in the human brain, saw that there are molecular changes of like hyperphosphorylated tau, which is what you see in neurodegenerative disorders. And using a inhibitor that they're trying to develop even for Alzheimer's disease, we could also reduce heroin-seeking behavior. We have a lot of ideas and a lot of results that can be practically implemented into treatment, into research projects. I just think that for substance use disorders, we need to have more medications based on science. And those are the toughest ones to get grants for. My fellow colleagues, it's very tough to write such grants. And so for me, I would spend a lot more energy in really trying to develop, as I said, using the science to guide more new novel medications, and also medications that are not addictive themselves. I think that we've learned a lot about the brain, and I think we need to move in that direction more.
Okay.
And also, I will say, I will do a lot of early with children, if we can get to kids, we can decrease significantly a lot of this.
Coylan has a question, which I'm not sure I understand. It says, what factors other than drug use affect cognitive performance in addiction?
Stress, huge, we talk about, especially coming back, to certain communities and so on. When you're living under constant stress, how can your cognitive abilities are not just a drug, toxins in our environment. A lot of kids we can see environmental toxins during development. It's huge impact on cognition later in life. So all of those things, absolutely. Yeah, negative environments are crucial killers of cognition.
I want to get a question from Goldie, who's asked a couple I didn't get in, but are changes with tobacco smoking also reversible?
Well, we do see epigenetic changes with tobacco. So, as again, I think a lot of these substance use disorders, we have told people that it's like the whole mantra, like once an alcoholic, always an alcoholic. And that may be for certain things because you're vulnerable, but the brain can recover. You're still vulnerable, but your brain can recover.
This David has a question. Do you know if addiction to prescription sleeping pills like Lunesta create permanent damage to the brain or can be reversed? Also, do you know if strategies to reverse the addiction to these types of drugs, I think they're called Z-class, differ from drugs like opioids or non-prescribed drugs? And how would it differ?
Yeah, I mean, I don't really know what a lot of the molecular changes with these are. I mean, clearly, these are depressants that we know can have long-term impact. But again, it comes back to dosing. A lot of people don't use the doses that are recommended, and but I can't speak about what specific mechanisms, unfortunately, as compared to the others.
Thank you so much. It's very kind of you. And thanks to Secret Science Club and Dr. Hurd, this has been absolutely fascinating. You mentioned in passing food addiction. How do you imagine that your research might impact that? Things like people craving sugar or people binging or even working with patients with diabetes. I mean, I'm sort of I have all these thoughts about it, but it would be amazingly helpful to help people regulate their eating habits.
Trust me, I need help myself. I definitely have a so, interestingly, when they give sugar to, we actually give sugar all the time to our rats. I mean, it's highly addictive, and they will self-administer sugar in very similar ways, and even to a greater extent than even cocaine. So companies know what to do to get people to like their products. You sprinkle a little, more and more sugar, and that's what happens. And we definitely see both molecular and in vivo neuroimaging changes similar to what you see even with chemical with substance use disorders. Yeah, clearly, the strategies for treatment are behavioral and replacement, and and even for substance use disorder, it's like trying to find a different, so compulsive behavior. Can you move that compulsion to something else that is healthier? And those are some of the behavioral strategies, even for food addiction. But food is also different. I mean, I have to say there's something extremely comforting, that I can't find a replacement for. But I'm gonna try.
Well, thank you so much for this talk and your wonderful generosity with our audience and all of our questions here. It's been wonderful